Excited today to have Dr. Paris Philippidis from Yale, and he's a translational physician scientist specializing in computational immunology, single cell genomics, and B cell biology in the context of infectious and inflammatory diseases. And he's currently a postdoctoral researcher at Yale School of Medicine in the lab of Dr. Stephen Kleinstein, and he's jointly mentored by a good friend and colleague of mine, Dr. Shelley Farhadian, who spoke earlier this year, and I think was trying to tell people that you are coming on later in the seminar season. And he's soon going to be actually transitioning to an independent physician scientist at Lausanne University Hospital in Switzerland. Sorry if I botched that. But so it's pretty exciting, right? He's been doing amazing things at Yale. He's getting ready to kind of launch his independent career, and but he's going to continue to work with all of us over here. So I am super excited today to hear about Dr. Philippidis' talk on B cells and the blood-brain interface, a legacy of immune dysregulation in treated HIV infection. Hi, Shelley. And that's his mentor. So just take it away.
So, hi everyone. Thank you so much, Leah, for the introduction. And also thank you for giving me the opportunity to talk about this topic, which is I'm really passionate about in general, B cells, infections of the central nervous system. And I think I really like this because as I was sharing earlier, B cells are actually often off the picture actually in HIV neuropathogenesis. And I hope today will show you that it's worth, pursuing this lead as well and study these cells more. So just a little bit of a background on neurocognitive impairment. So neurocognitive impairment in people with HIV is as old as the pandemic itself. And in fact, as you can see in this timeline, several events and discoveries have happened over time as the pandemic evolved. But the most decisive moment in this history of neurocognitive impairment was the introduction of antiretroviral therapy, or ART, which currently induces a durable suppression of HIV replication and actually brings the life expectancy of treated people with HIV close to that of the general population. But the thing is that despite this very effective antiretroviral therapy, a subset of people with HIV still present with cognitive complications, and in most times they have mild to moderate cognitive impairment and more rarely severe neurological disease. So we are currently in the era of sustained inflammation as one of the major mechanisms of underlying cognitive impairment in people with HIV who are treated. And this is what actually the research community tries to further, like this persistent inflammation is tried to be further explored, both on the viral end and also on the host immune response end. And also, I just added a note down here showing that like a very previously popular classification system for these disorders was HAND, or HIV-associated neurocognitive disorders. But this now tends to be replaced by what is called HIV-associated brain injury, or HABI, which more captures more the mechanistic actually basis of cognitive impairment in people with HIV. It doesn't solely rely on a cognitive score, which may be subject to a lot of confounders. So when looking at HIV neuropathogenesis, two major cell populations are well established as being important or mediating at the establishment of an HIV reservoir in the brain and immune responses. So first, there are these monocytes which are infected by HIV and are thought to be the Trojan horse, which brings the virus into the central nervous system. And these cells after further differentiate into macrophages. And another myeloid cell in the brain are microglia cells, which are basically the brain resident immune cells, which can also be infected and also mediate neuroinflammation. So that's on the myeloid end. And then on the adaptive immune response end, there are T cells, which are shown here as these green cells that are the major, or like, let's say, the main target of the virus. And they also can migrate from the periphery into the central nervous system, both sustaining the reservoir, but also mediating inflammation. But what is not on this picture, and actually is not in any of the HIV neuropathogenesis figures, are B cells. So what is actually the B cell component in this cascade? And before I show you a bit more on B cells in the context of HIV, I just wanted to acknowledge that B cells are rare in the healthy human central nervous system, or CNS, and mainly found around perivascular spaces, the meninges, and also in the cerebrospinal fluid. And they have mostly been studied in the context of acute or chronic neuroinflammatory diseases, but in the context of overt neuroinflammation. And I'm showing some of the conditions here, like multiple sclerosis, neuromyelitis optica, and glioblastoma. And what is also important to note is that, as shown in this figure here on the left, B cells can have both anti-inflammatory or neuroprotective functions in the brain, or they can have pro-inflammatory functions. So there is this balance between protective and detrimental effects, and that underlines the fact that they have to be studied in each specific condition or even disease state separately. So now, looking at the HIV like context, in viremic HIV infections, there have been B cell changes are very well established, and are numerous. Some of them are shown here, including polyclonal activation and hypergammaglobulinemia, also exhaustion and increased B cell turnover, and in some individuals, even the development of broadly neutralizing antibodies. And so these changes may occur in almost all B cell developmental stages, as shown in this figure on the right. And for example, there have been reports showing an increased immature transitional B cells in association with the T cell lymphopenia induced by HIV, increased short-lived plasma blasts as well in the presence of HIV viremia, and also a decrease in resting memory B cells. But this is for viremic infection. But what is even more interesting is that while on therapy, so after the virus has been suppressed, almost all of these B cell changes are reversed, but there is still a reduced frequency in some cases, a reduced frequency of class unswitched or IgM positive memory B cells. And this is interesting because this is a change or a deficit that has also been described in splenectomized patients in whom we observe a loss of spleen-derived marginal zone B cells. And this is also this also provides insights into the fact that people with HIV have also increased susceptibility to pathogens that are treated by T cell independent B cell responses, such as pneumococcal infections, which are also very common and may be very dangerous in splenectomized patients. So now, what about the central nervous system? So as I said at the beginning of the presentation, humoral immunity remains a black box in HIV, especially treated HIV infection. But what we do know about humoral immunity is that, for example, the CXCL13 B cell chemoattractant is associated with inflammatory biomarkers and also neurocognitive impairment. And also, people with HIV may have an increased intrathecal immunoglobulin G synthesis, which also correlates with blood-brain barrier dysfunction and also neurocognitive impairment. Also, the CSF antibodies are different from the ones in the plasma, and this goes for their function, which may be reduced in the CSF, but also the antigen binding patterns. And last but not least, these antibodies are not only HIV specific and can also be they can also target co-infections or latent pathogens such as the Epstein-Barr virus, um, the anti-EBV IgG antibodies being associated with neurocognitive impairment as well, which actually suggests that there may be polyspecific or bystander responses on top of HIV-specific responses. And similarly, autoantibodies are also can also be present. A typical example are anti-CD4, but also other brain self-antigen have been found as targets of autoantibodies in the CNS, especially in the context of viral escape. So looking at this indirect, actually, evidence, the general hypothesis is that intrathecal B-cell responses, whether HIV specific or not, may contribute to persistent neuroinflammation even in the presence of treatment, effective treatment with suppressed plasma viremia. So we asked this very simple question: what is the role of B cells in HIV neuropathogenesis during viral suppression? And to answer this question, we leverage the Yale HAR cohort, which enrolls people with HIV. In this case, all of them were virally suppressed and neuroasymptomatic, and also people without HIV demographically matched. Here in these boxes here, I'm showing some very high-level demographics, but what I wanted to highlight is that people with HIV were virally suppressed for a median of 16 years, some of them even more, and they had a pretty good viral control with no rebounds, for example. They were really in sustained viral suppression over all these years. And all of them were treated based with a regimen including an integrase inhibitor. So these participants underwent cognitive assessment as well as a high-volume lumbar puncture and blood draw. And these samples and data were analyzed in three different study arms. So the first study arm included 7 people with HIV and 6 people without HIV, whose samples were analyzed using the 10x genomics 5' VDJ single-cell RNA-seq pipeline, which provides information on gene expression, but more importantly, on B-cell receptor repertoire. The second study arm consisted of 7 people with HIV and 6 people without HIV, who were different than the first arm. And their samples were analyzed this time using the 10x genomics multiome pipeline, single nucleus, which in addition to gene expression, also yields information on chromatin accessibility. And the last study arm of CSF studies included the broader HAR cohort, 44 people with HIV and 22 controls. This time, the supernatant, CSF supernatant, which was processed using a multiplex cytokine panel for proteomic analysis. And so, in addition to the CSF, we also studied choroid plexus tissue and adjacent brain parenchyma from 3 people with HIV and 3 people without HIV who were enrolled in the National NeuroHIV Tissue Consortium. These were people with HIV were also recently suppressed, and after dissociating the cells from the brain tissue, these were further analyzed using the 10x genomics single nucleus RNA-seq pipeline. So looking at the CSF results, the very first conclusion we can make at high level is that it is possible to profile CSF cells in the absence of overt neuroinflammation. And I'm saying this because the CSF of these people, of people with HIV, in this case was actually pretty much the same as the CSF in controls, macroscopically at least, including less than 5 cells per microliter. So we are able to profile them regardless. And I'm showing these two UMAPs comparing blood immune profiles with CSF immune profiles and showing that we are able to identify the main immune subsets in both compartments, including T cells, myeloid cells, and B cells. And I will be focusing on B cells for the rest of the presentation. So now zooming into this B cell compartment, we further subcluster these cells into 4 different subtypes, including naive B cells, resting in atypical memory B cells, and also plasmablasts. And as you can see in these box plots in the middle, the frequencies were comparable between people with and without HIV. Although I have to say that these cells, B cells were really rare in the CSF, accounting for 0.27% of all cells. So really low number of cells, but it is already a finding that we find them at all in the absence of, neuroinflammation, or like at least macroscopic or overt neuroinflammation. So another observation here is that the composition of CSF and PBMCs or blood is different in terms of B cell subset, with CSF being dominated, almost exclusively composed of resting memory B cells, whereas blood included more naive cells. So the fact that we find any B cells at all in the CSF suggests that they may play a role in CSF immunosurveillance and homeostasis. And although they are quantitatively comparable between people with and without HIV, we were wondering whether that's also the case for their function, or in this case, transcriptomic profile. So to answer that, we leverage the RNA-seq component of our data. And here I'm showing 2 principal component analysis plots in which each dot represents an individual and summarizes basically all the transcriptome of this individual's memory B cells. And light green is people without HIV, light purple is people with HIV. And as you can see here on the right, memory B cells in the CSF were pretty distinct, actually, between people with and without HIV, which was not necessarily the case for blood. So now that we've seen that there's a big difference in the transcriptomic profile, we wonder what these differences reflect in terms of biological processes. And to further answer this question, we performed differential gene expression and pathway analysis. And here I'm showing the results of this analysis. Each row represents a biological pathway. And for simplicity, I kind of group these pathways into 4 different categories, consisting of antigen presentation, interferon type 1 and 2 signaling, cytokine signaling, and more broadly viral responses. So as you can see in this plot, there's in CSF group 1, the first column here, these impairment, all of these pathways are downregulated. And while we discovered these changes in CSF group 1, which I remind you is the first study arm, we were able to validate them independently in this CSF group 2, which was the second study arm. So we basically found the same thing in these 2 data sets. And despite the fact that, in addition to the fact that these were 2 different groups of participants, I think what makes it even more robust is that these were 2 very different experimental protocols as well. So in the first case, we look at cells. In the second case, nuclei, and these 2, and the processing of the tissue is also different. So this further consolidates, I think, the fact that these cells are impaired. So we are based on these results, I think we can say that there is a compartmentalized immune dysfunction in CSF memory B cells of virally suppressed people with HIV. And the next logical question is whether these changes are reflected also at the epigenetic level. So to answer these 2 to address this question, we looked at the chromatin accessibility component of the second study arm, and this is work done by Chang Lu, a PhD student here at Yale. And here on top, you can see a UMAP, which basically shows clustering of the cell subtypes that we identified based on the RNA data. But this time, the clustering was done based on the chromatin accessibility features. And that what this basically tells us that here, down here, you can see in light green, I hope you can see this light green cluster, which represents B cells, and tells us that these B cells have also distinct chromatin accessibility profiles compared to other cell types. And the same thing is shown in this heat map down here, where you can see this group of peaks or chromatin accessibility peaks that are specific to B cells. And now to look at whether these changes I showed at the transcriptomic level are connected with the epigenetic features, we explored several loci that are involved in the processes that were impaired, such as antigen presentation interference signaling. And here I'm showing only an example of the HLA-DQA1 locus involved both in antigen presentation process and also interference signaling. And what you can appreciate here is these peaks represent chromatin accessibility, and on top you can see people with HIV, and the bottom row is controls. So as you can see, there is a higher chromatin accessibility in people with HIV, both at the transcription starting site of this locus, but also in neighboring chromatin, in neighboring genomic regions, such as the one in this blue box, which has significant peak to gene links, these blue lines you see here, which basically means that these chromatin accessibility changes impact meaningfully the expression of this gene. So this provides like a level of evidence that epigenetic regulation may also contribute to the transcriptomic alterations, although we would need larger scale data to make definitive conclusions about that. So now moving at the proteomic level, so the third study arm for CSF studies, we also looked at cytokines and chemokines within the CSF. And for that, I would like to just highlight that this was done at the bulk level. So we're looking at the CSF proteomics that represents all of the cell types, not only B cells. This is in supernatant. And so when looking at these chemokines and cytokines, we observed several cytokines that are associated with B cell biology. And again, this doesn't mean that these are produced by B cells. They may be produced by T cells, by myeloid cells, but they favor B cell differentiation in the first case here on the left, and also B cell trafficking and homing. And you may recognize some of them as being involved in specifically NeuroHIV, such as IL-10, CXCL10, CXL12, CXL9, etc. So although we cannot say that this originates from B cells, this suggests that there is the CSF environment is B cell supportive and may promote, all these processes that I showed before at the B cell level. All right, now shifting a bit gears and looking at antigen experience and tissue homing. So far, we've looked at the, let's say, functional properties, or which are represented by transcriptomics. But in general, memory B cells are thought to migrate from the peripheral blood into the CNS in response to chemotactic signals. And to do so, they have to go through restrictive barriers such as the choroid plexus and the meninges. And while it is quite well established how these B cells are recruited into the CNS during inflammation, that is in an antigen-dependent manner, it is unclear how B cell migration and trafficking happens in the absence of overt inflammation, especially in a context where it's not a healthy state, a perfectly healthy state, like such as in controls, but it's like healthy enough, like in people with HIV who have a viral suppression and no macroscopic inflammation. So to answer this question, we complemented the transcriptomic data, which I remind you showed compartment-specific pathway level dysfunction in memory B cells with single-cell B cell receptor sequencing data, which add or offer insights into clonal architecture, tissue trafficking, and antigen exposure history. And I will be first showing some results regarding the compartment differences. So these pie charts here show the isotype frequencies in both CSF and blood. And by the most common isotypes are IgG, IgM, IgA, and IgD. And so, as you can appreciate on the left pie chart representing CSF, most of the isotypes, the cells that were bearing B cell receptors were IgG positive. So more than 50% of the B cells were bearing an IgG receptor, followed by IgM, 1/3 of the cases, and then IgA. And in blood, however, B cells mainly expressed IgM isotypes, followed by, again, IgG and IgA. So I wanted to remind you that these are not soluble antibodies. These are B cells that are carrying a B cell receptor, which will, if an antigen is present, then may differentiate into antibody producing cells. But these are not secreted antibodies, which is actually one of the main differences with the currently available literature, like looking at intrathecal soluble antibody production. And then looking at expansion rates, I won't spend too much time on this, but this basically shows the number of expanded B cells in the CSF and blood. The top row shows blood, the bottom row CSF, and the left column shows people without HIV, the right one, people with HIV. And the red, like the height of the bar, shows the number of different BCRs we found or clonotypes, and the red highlighted area shows how many of them were expanded. And basically the percentage summarizes this expansion rate. So what we can conclude from this figure is that basically there are no major differences in clonal expansion between people with and without HIV, which is, I think, corroborates the transcriptomic findings that quantitatively these B cells do not seem to be very different in between the two groups. And so, yeah, that's summarizing exactly what I just said, that there are differences across compartments, but not really differences between people with and without HIV, at least in terms of clonal expansion. But there's more than clonal expansion in B cells. And in fact, one of the major processes that B cell receptors undergo is called somatic hypermutation. And this is a hallmark. It's as a hallmark of antigen-driven B cell activation. And here I'm just showing, for those who are less familiar, how this process very briefly works. So it's depicting a lymph node here, and these red dots represent an antigen which enters the lymph node and comes into contact or is bound by naive B cells. And once this interaction happens, these naive B cells migrate into the germinal center of the lymph node. And so here you can see that after they've entered this so-called dark zone, they start proliferating and inserting mutations in their receptors, B cell receptors. And the purpose of this process is to make the receptors increase their affinity as much as possible, so that they bind very nicely and smoothly the antigen, and therefore can better respond to this antigen. So once this process is finished, these B cells then are tested against the given antigen. And if their affinity is good enough, they may further differentiate into memory B cells or plasmablasts. And if their affinity is not good enough, they undergo apoptosis. And this is a process called positive selection. And the entire process I just described is called affinity maturation. So why I'm saying that is because we are able to computationally infer the somatic hypermutation of the B cells we're looking at. And this is what we did in this case. So um, this is a UMAP plot showing the four subsets I showed previously, but this time overlaying somatic hypermutation rates. And the darker the color, the higher the rate. So as expected, these naive cells are unmutated, which makes sense because these are antigen naive. Whereas antigen experienced cells such as memory cells, memory B cells, and plasmablasts have higher mutation frequency rates, darker colors. And there's also differences across isotypes. So IgG and IgA are class switched isotypes, so more experienced cells, and therefore higher mutation rates compared to IgM who have lower rates and IgD who have no mutations. But what is even more interesting is that when comparing people with and without HIV in terms of somatic hypermutation, which I remind you reflects antigen experience and also clone selection, what we saw is that in the blood compartment, people with HIV had lower mutation frequency than people without HIV in memory B cells. And the difference was pretty striking. So if we think the hypothesis that is currently proposed is that B cells and antibodies are passively transferred from blood. So if that was the case, if B cells were just patrolling the CSF and just passively transferred from blood, we would expect that these difference in somatic hypermutation is reflected also in the CSF if the B cells in the CSF reflect the blood B cells. But that doesn't seem to be the case. So this difference was not observed in the CSF. And in fact, we calculated the gradient of mutation frequency between the CSF and blood compartment depicted by this difference here. And we were struck by the fact that people with HIV actually have higher mutation frequency in the CSF, despite the fact that they have lower frequencies in blood. And so 2% more mutation in the CSF compared to controls who have 2% less. So this difference, which is pretty substantial, makes us think that there is a recruitment of antigen experienced clones into the CNS, or even local affinity maturation, both of which point to a persistent antigenic stimulation. And so here I'm just highlighting that this doesn't exclude or preclude the hypothesis that most of the soluble antibodies are transferred passively. Probably both cases or both hypotheses are true at the same time. But one looks at the soluble antibodies, and our project right now looks at cells. So despite antiretroviral therapy, HIV infection differentially impacts B cell maturation across compartments. And so now zooming out from the molecular findings, we also wanted to look at clinical outcomes, and in this case, neurocognitive performance. And to do so, we were very happy to have help from Dr. Leah Rubin, who actually used a data-driven approach, leveraging cognitive assessments from the larger cohort, in this case, almost 100 individuals. And by doing so, Dr. Rubin was able to reduce 16 individual cognitive test scores into 3 orthogonal factors. And here I'm showing the results of the entire cohort. On top, you can see the cognitive domains. This is a data-driven approach. So these are the cognitive domains that were found, that showed up by summarizing these cognitive assessments. And the 3 ones that were found was processing speed and executive function, motor function, verbal episodic memory. And as you can see, the factor score 1, representing processing speed and executive function, was found to be lower. The performance was lower compared to lower in people with HIV compared to people without HIV. So, which is actually consistent with what is known by the current literature in NeuroHIV. So looking at these results, we then we then asked the question whether this difference or this poorer performance is somehow connected to the molecular finding I just showed in the previous slide, the difference in somatic hypermutation between CSF and blood. And in fact, the answer is yes. And here I'm showing a scatter plot calculating the correlation between the somatic hypermutation difference and the factor score 1 representing processing speed and executive function. And this shows that as this difference between of mutation frequency increases between blood and CSF, the performance, cognitive performance in this domain reduces. And this difference remains significant after adjusting for age and education. So there may be this like molecular impairment, or this difference in somatic hypermutation may underlie cognitive performance or cognitive impairment. But as you know, cerebrospinal fluid is not brain. So we also wanted to take a closer look at the brain parenchyma. And this was only made possible in collaboration with Michael Corley, who did a wonderful job in dissociating cells from brain parenchyma choroid plexus. And here I'm showing a U map of the cells we identified. And in this case, we focused on B cells again, subclustered them, observed again the same separation between plasma cells and B cells. And the principal component analysis here again showed a very big difference between B cells in people with HIV and people without HIV. And following the same approach as we did for CSF, we also did pathway analysis. And not surprisingly, we found the same pathways that were impaired in CSF were also impaired in the choroid plexus. So we believe that this transcriptional reprogramming of B cells is shared between the two compartments. So collectively, looking at these results, we propose a model in which the reduced antigen presentation capacities and also interference cytokine signaling and responsiveness of B cells, memory B cells, favors persistent antigenic stimulation despite viral suppression, and this further fuels actually ongoing affinity maturation and B-cell somatic hypermutation, or the recruitment of more mutated clones into the CNS. And the next, now the question is what these B cells react against. So, the obvious answer would be HIV, but it's also possible that these receptors are specific against opportunistic pathogens or even self-antigens, brain antigens that may be produced as part of bystander neuronal injury. So to summarize and conclude the presentation, we showed that there is a compartment-specific legacy remodeling in CNS memory B cells, and that is despite long-term viral suppression on antiretroviral therapy, and also that the affinity maturation within the CNS may be impaired or reflect antigen-driven selection of specific clones, and the molecular feature of these changes may underlie cognitive impairment in ART-suppressed people with HIV. And last but not least, these differences, at least at the transcriptomic level, seem to be shared between CSF and brain. And with that, I'd like to first and foremost thank tissue donors and participants in this study, and of course, my two wonderful mentors, Dr. Shelley Farhadian and Dr. Stephen Kleinstein, and all these people who made this project possible. And yeah, with that, I'd be happy to take any questions and discuss these results.
Thank you so much for an amazing presentation. That was awesome. I see Shelley here now too. That's amazing. I will go to the chat first. Scott, do you want to ask your questions? Dr. Latender?
Hi, Paris.
Hi, Scott.
I keep seeing more and more of your data, and it keeps getting more impressive and beautiful. So congratulations on all of this work.
Thank you.
Yeah, my questions just had to do with just thinking through, sources of uh, potential confounding of the data. So sorry for for that. But
Of course.
one had to do with who the people without HIV were, and what herpes virus infections, EBV and others that they had compared with the people with HIV, if you know that. And then the other just had to do with the cause of death for the choroid plexus sampling. If you know that for the people with HIV and people without HIV, I'm sure it's known, but I would think that the causes of death would differ between people with HIV and without HIV, and there's probably not enough a large enough sample to do a to be powered to do any
Okay.
sort of test, but it's valuable still for us to think about, what causes of death may potentially have influenced the results.
Yeah, thank you for these questions. To the first part, unfortunately, I don't have the profile like like at least herpes viruses, CMV, EBV. We do have. We actually were planning to look at these profiles. We haven't done this for this project. We were hoping to do this as a comprehensive analysis with CSF polyclonal profile that you've already heard about. So. This will, I think we will follow up with these questions, but I think it's important, and we have actually uh, already discussed in trying to think how we could even look at the latency in the brain of these viruses, because like specifically in brain tissue, because it's this could answer many questions, and actually the profiles are very different. This cannot confound, they're still right, but unfortunately I cannot answer. I don't have
And
this information so far yet.
viral hepatitides would be important too, particularly hepatitis C, which I think is all
Right.
very B cell stimulatory. And so were the people from, I'm sorry, I missed it. Were they from Max and Wise or
Uh,
no?
So you mean the cohort, or
Yeah, the people without HIV.
so they were all including the HARC cohort, like the Yale HARC cohort.
Okay. All right. All
Yeah.
right. Sorry for missing that.
So
Okay.
and I'm sure we have this information for most of people. We just haven't analyzed it yet. Yeah.
Okay. All right. And
And so to
then,
your second point, um, it is, I know it will sound surprising, but we don't really have yet the exact cause of death. We have tried a lot to obtain this information. We haven't been successful so far. What I can say is that we have the biopsy, brain biopsy results. And at least at the CNS level, at the brain level, we most like if not all of them actually had no like brain pathology or just age-related neurodegenerative like non-specific lesions. But we unfortunately don't have the cause of death, and we have struggled a bit with obtaining this information. I don't even know why it wasn't part of the data we received, but we're trying to recover this information now.
well, you and Dr. Farhadian are both extremely brilliant and accomplished, and so I know you'll look at postmortem time as well. So
Absolutely, we did that. Yeah. So the postmortem time is already we will report that, and it's it's variable, but it varies between 1 and 4 hours.
okay, great,
Yeah.
wonderful. All right. Well, thank you for letting me go first, Leah.
Of course. There is somebody else, Arpan. I think that there's a
Yeah, and while we're looking, I would just wanted to also say to Scott and everyone that these people with HIV and without were very well matched. Although, we don't necessarily report all of their comorbidities, we didn't necessarily match them beforehand for everything. After, like post-hoc comparisons were really, really balanced groups. Yeah.
Hi, Paris. Wonderful work. So I have a quite technical question. So as you know that the percentage of B cells in the CSF is quite low. So exactly means what is the absolute number of minimum B cells you captured per patient to do all this detailed analysis of the attack-seq as well as the B cell receptor analysis and the hypermutations, because we do some of the similar type of work in our rhesus macaque models in the context of substance of abuse, but we able to collect very less number of cells, and this is very difficult to do. That's why I wonder what is in your case.
Yeah, it's a wonderful question, and I'm so happy you asked this because I think not many people think about it, and I think this is very important. And a lot of my energy went into consolidating the results just because we had a few B cells. So the let's say the median number of B cells was 5 to 10. So it's like very few B cells. You really are right. Um, so it's not surprising that in your case you didn't have enough. I think this is expected, right? Because these
Yeah.
people do not, as I was saying in the beginning, they don't have overt neuroinflammation. They don't have these increased signals that bring B cells in. Um, However, I do think we can still analyze them. And in our case, what we were discussing is that we can't do much about how many B cells are in the healthy CSF, right? We can't really change that other than like increasing the sensitivity of the current analysis, which may take some time. What we can do is ensure consistency. So if we cannot be sure that these 5 B cells represent a significant change, we can say if this difference is present across all individuals. And the more patients we have, the more we can ensure consistency. And in our case, these results were consistent across all individuals. So even though they had few B cells, if all of these B cells, even if they're few, have the same changes, that makes us think that maybe this is a true signal. And actually, this is the best we can do right now. And also, I didn't show all of these results, but I ran a lot of rounds of validation and different orthogonal ways, computational ways of validating these transcriptomic results, including like accounting for outliers, looking for outliers, accounting for them if they're present, they weren't present in our case, calculating gene scores, like a lot of different ways of like making sure what I'm showing is actually true, at least in these data sets. So I would encourage you to, and that was my first like discussion with Shelley when we started this project. Let's look at the B cells regardless, even if quantitatively they are the same and low numbers, because I think this also offers a framework for studying, as you said, quiescent pathologies. In the case like other latent infections, for example, or like cases where, for example, people with multiple sclerosis who are not in a flare, right? So they may not have any cell at all. That doesn't mean that their B cells are normal just because they're numerically the same with controls. But yeah, thank you so much for asking this. That was a very real headache, and in my case, really a challenge. And I really wanted to make sure that it's taken
And
care of.
another clarification, you did the fresh capture or you did frozen cells after
It
cut?
was Yeah, it was a fresh CSF, and actually, I think an important fact, and thanks for bringing this up also, is that in order to have this number of B cells, I think other groups didn't have even that many B cells. They had none or like one. So I think it's also important to look at the volume of the CSF that is collected. So if we had like an active, acute neuroencephalitis, like meningoencephalitis, for example, we wouldn't need as much CSF because there's so many cells. But in this case, it's normal CSF. So we will need high volume CSF in order to be able to identify these cells. And that also goes for other potentially rare cells or cell states. So, CSF volume is really important in this case. You're right. And it was fresh. Like
Sure.
within 1 hour of collection, it was processed.
Yeah. Thank you.
Of course.
And I see Scott has another question. Cecilia, did you have one? Because I see you popped on just to make sure.
I'm just here because I think it's polite to show that I'm here. But
I
yeah,
love it.
I did have a comment that what you call antigen experience cell is based on the phenotype. I don't know if I blinked in that moment and I missed it, but you don't yet have the information about the actual clonal specificity in terms of HIV or herpes virus or whatever. So the comment would be in relation to the ratio between specific cells and bystander, and how that relates with the cognition and other neurological outcomes. Just thinking of a parallel with CD8 cells, that at least in the macaques, we see that kind of thing.
Yeah, great point. And actually, I think that's, at least in my eyes and in B cell biology, that's the big elephant in the room, right? So, are these B cells actually HIV related or like specific or not? And that's the next step. I tried to infer them computationally, but as you may know, this is often it has a poor performance. The inference methods and comparisons we currently use are not great. So what we did is we actually followed up on this work and submitted an R21 grant, which was accepted to make BCR-derived monoclonal antibodies. And we're planning to test them against over 1 million actually antigens, including, I think, 7,500 HIV epitopes, linear epitopes, also self-antigens, bystander, co-infections, opportunistic pathogens. And we also, depending on how this goes, we may add like a conformational layer, so also conformational binding to increase sensitivity. But you're absolutely right. And I think that either way, even like whether these are HIV specific or not, I think it's interesting to look at the patterns. And that brings the subject back to Scott's question, mainly saying that maybe these are not HIV specific, but their responses against opportunistic pathogens may give us more insights on why these people have like persistent cognitive impairment. So that's a really great question. And if these are HIV specific, I think another question that comes next is, what is the breadth of HIV epitopes?
Yeah.
Because if like this is restricted to a few epitopes, this may even be key therapeutic, diagnostic or therapeutic targets. Even if the breadth is higher, then we might think differently of how these responses work in the CNS, especially given the viral particle or viral protein persistence that is thought to be.
I don't know the details of the technology, but if you have the sequence of the FAB hypervariable region, there is a database, I think it's Alex Setti here in De La Jolla Allergy, and he has this huge database that you can computationally try to dock with your hypervariable region. I think it might be interesting for a student there to play with this.
So I did that. I did compare it with publicly available sequences. Specifically, I used the IEDB, it's called, database, which is the most like well-established database and validated. And there was no match, but there's still like there's so many considerations, what criteria you use for the match, how close you want it to be, because of the how different the B cells are across individuals. It makes it very tricky to match the exact sequence. We do this at the amino acid level, but even one amino acid can completely change and shift antigen binding. So it's very challenging. And we're doing a lot of B cell work in our lab, and it's rare that we actually find reliable matches with publicly available data sets, at least with that few B cells.
You really need the BLCLs to do it, right? Yeah.
Exactly. Yeah. Yeah.
Wonderful. Thank you. Thank you so much.
Thank you so much. Thanks for your question.
Marcus?
Hi, yeah, thanks, yeah, and thanks, Bryce. Wonderful work, really, really interesting. And following a little bit on what Scott asked and what also Cecilia went to, I'm wondering, and this is in general, also in the context of neurodegeneration, do you know what the vaccination status of the people was? Because, how would that, you would wonder, I mean, that would influence, B cells probably, particularly in a certain way. And I was just wondering if you have looked at that.
Yeah, we haven't looked at that, but this is something we were actually even discussing 2 days ago with Shelley. How could we incorporate this, vaccination more broadly, also with regards to treatment? We haven't done this yet, but I think that it would be great to look at this as well, and also look at the correlation, like the associations, at least with the molecular findings.
Yeah, I mean, you would expect, depending on how they were vaccinated, right? You would expect that they have antibodies against certain antigens, right? If you would for the profile. But it would still be interesting to see between, people with HIV without HIV, right? How this compares, right? And then how this perhaps influence, how many B cells you actually get all over, right?
Right. Right, and I think that's that also nicely fits into a project that we're proposing and trying to start, is looking at polyclonal antibody responses as well, right? Which will better reflect at least or either past exposures to pathogens, but also vaccination, and then look at the association with cognitive outcomes and even molecular differences at the single cell level. Yeah.
Cool. Thank you.
Thanks for the question, of course.
Very nice work. Very nice work.
This was
Thank
an
you.
amazing discussion. Does anybody have any last minute question? This was, again, Paris, thank you so much for coming and speaking at the seminar. Amazing presentation, great question and answer. Can't wait to see what you keep doing both over, abroad as well as in the US. Not Shelly, Caroline, speaker for next week.
Yes. So next week we have Rebecca Veenhuis from Johns Hopkins School of Medicine, and her talk is Redefining HIV Persistence, the Role of Monocytes and Macrophages as a Latent Reservoir. So
awesome.
We hope
then. To see everybody next week. And again, thank you guys again for coming today.
Thank you so much.
Thank you.
Bye,
Bye.
everyone.